as I discussed that in the coming videos, we'll be talking about few questions related to life sciences. The questions for today are, the supramolecular complexes of proteins, nucleic acids are not held by. We should be clear about the wordings in such questions. Like here, the important point to be noticed here is not held by. So we know that these complexes, these complex structures, that is the secondary and the quaternary structures, they are held together by weak interactions, which include the ionic interactions, the hydrogen bonds and the hydrophobic interactions. Those which are not involved in supramolecular complexes, they may be involved in the primary structures but not involved in the secondary and tertiary structures are the covalent bond. So answer here is the covalent bonds, right? Second question, it states that based on the HH equation, that is the Henderson and Hasselbach equation, which we generally use for calculation related to buffers, calculate the pH when the acetic acid is half dissociated to acetate. So we are talking about the acetate buffer here. They have given the dissociation constant of the acetic acid, that is pKa is 4.76. By definition of HH equation, we know that pH is equal to pKa plus log of A minus, that is the H plus acceptor, and upon log of HA, that is the H plus donor. So here it says that acetic acid is half dissociated. That means if you start with 1 molar, you have 0.5 of acetic and 0.5 of acetate. If you start with 2, you have 1 of acetic and 1 of acetate. That means there is an equal concentration of the proton donor as well as the proton acceptor. So here we have this ratio is equal to 1. So solving this, we get that pH is equal to pKa plus log of 1. By definition, now we know that log of 1 is 0. pKa is given 4.76. So pH is equal to 4.76, right? Because we know that this log of 1 is equal to 0. So therefore, the pH when acetic acid is half dissociated to acetate is 4.76. Rather, this defines pKa. We define pKa as a pH where the acid is half dissociated. Thank you.